this is another exercise that's dealing with the enterics. Specifically, this test is what is known as the TSI. Just as a refresher, the enterics are your gram-negative non-swarming bacilli. They're commonly found in the intestinal tract. So therefore, if you find them or detect them, it's indicative usually of fecal contamination. So what is a TSI? It stands for triple sugar iron. The three sugars that are present are glucose, lactose, and sucrose. There is a pH indicator added. The pH indicator is phenol red, which we have seen multiple times before. Remember, neutral or alkaline is going to be red, and it is yellow when it's acidic pH under 7. This too is grown as or made as a slant, and when you inoculate it, you're going to do something a little bit different. You're used to inoculating on the slant, just spreading the inoculum or the sample on the slant. But on a TSI tube, you are also going to inoculate the bottom part of the tube, which is what we call the butt. So you're going to stab the butt and then just spread along the slant. What you're looking for here is on the slant, obviously it's exposed to atmospheric air, so any growth there is going to be aerobic. The butt, when you inoculate that, if something grows in there, that is a more anaerobic environment, so that's what you're actually looking for, is anaerobic growth. There is also iron added to this media, and the end result, you're going to be looking for hydrogen sulfide production. So there's a lot to interpret from this one tube. You would inoculate it, as I said, inoculate the slant, stab the butt, incubate it. After the incubation period, you're going to pull it out and you're going to look at it. Now, the very first thing you're going to see, do you see black anywhere in the media? If so, that is positive for hydrogen sulfide production. Look to see, is there growth on the slant portion? If there is, that's positive for aerobic growth. Is there growth deep in the butt section? That would be anaerobic growth. If only the butt, only the bottom part of it is yellow and the slant is still red, that means that only one of those sugars was fermented. If the entire tube is yellow, then that means that at least 2 of the sugars were fermented. Do you know which ones? Because remember, there's 3 in there. No, but if the entire tube is yellow, then you know that at least 2 of the sugars, was fermented. If the entire tube is red, or kind of orangish color, then none of the carbohydrates were fermented. You didn't produce any acid at all. If there are cracks within the agar, or sometimes I've seen where at the very bottom of the tube, the agar has actually literally been pushed up, maybe about I've seen it as much as an inch. So any movement of that agar, any cracks in it, any bubbles in it is indicating that gas production occurred. Because remember, you're looking at carbohydrate fermentation as one of the types of tests here, and one of the results or byproducts of fermentation potentially is gas production in addition to the acid production. So in this series of pictures, it shows some examples of potential results that you might see. Over on this one, you can definitely see that there was some hydrogen sulfide produced, that you see the black there. Looks like the bud is still alkaline and the slant is slightly acidic. You do record the 2 separate results when you're recording them. You record the alkaline as ALK, acid is recorded as A, gas is recorded as G. And so you have to record the butt versus the slant. This one is negative and negative. This is acid, acid. This is alkaline, alkaline. Over here, you definitely had the hydrogen sulfide production quite extensively in here. In the butt, which indicates it's more anaerobic, you did have growth up here on the slant which also means you had aerobic growth, but it's interesting that the hydrogen sulfide production is under the anaerobic conditions. Over here on this last tube, it shows a very good example. You can kind of see that there's growth up here on the slant, but you can see here where the auger is actually split in half down here in the butt section because of the gas production. So you need to look at these tubes very carefully. Sometimes the gas production is not this dramatic, so you need to look at them very carefully. The hydrogen sulfide is pretty easy to see because of the black. Look for the color differences. Look at is the growth on just the slant or just the butt or on both. And then look, sometimes the gas is just like little bubbles embedded in the auger. So once again, testing your observational skills of what do you see. Make sure you record all those results. So this is the TSI triple sugar iron test for Enterics.